Assalamu alaikum students. How are you? I hope you are all fine and enjoying the study at your homes. Today we are going to start our lecture number 5. And lecture number 5 have its connection with the lecture number 4. If you remember that in lecture number 4 we have studied the water balance problem. We have observed that what happens when we placed an animal or a plant cell in different types of solutions. Either the solution is hypertonic, hypotonic or isotonic solutions. So in our previous lecture, we have studied the condition of a cell when we placed it in hypotonic, in hypertonic or in isotonic solution. Now in this lecture, we will see that what is the connection between osmosis and plant cells. Students, recall the definition of osmosis. Basically, osmosis is the movement of water across a semi-permeable membrane from a solution of lesser solute concentration to a solution of higher solute concentration. What is semi-permeable membrane? Semi-permeable membrane is those which allows smaller molecules to pass through it, but the larger molecules cannot pass through this membrane. And the solution which have lesser solute concentration is called hypotonic solution, and the solution which has higher solute concentration is called hypertonic solution. Now we are going to observe the phenomena of osmosis in plant cells. Basically in plants, the surface of leaves is covered with small pores that are called stomata, and that pores are responsible for the exchange of gases in plant cells, and that pores are guarded by guard cells. Stomata are basically the openings in leaf epidermis and are surrounded by guard cells. And you know that during daytime the process of photosynthesis is happened, and in this process plants can make plants can make glucose with the process of photosynthesis. So that during daytime guard cells are making glucose and so are hypertonic. So better recall that hypertonic is that solution which have higher solute concentration. Okay, one will make a solution. We have two major component. One is solvent and the other one is solute. So during daytime when plants are making glucose, so they are hypertonic because they have higher concentration of glucose than their nearby cells, so that the water enters in the guard cells and they are swelled. In this form, they assume a rigid board shape and a pore is created between them, and that pore is called stomata. But the condition is opposite at the night time. At night when there is low solute concentration in guard cells, water leaves them. At night there is no process of photosynthesis because there is no sunlight. So that the process of photosynthesis stops and plants will not make the glucose. And due to this reason, they become hypotonic. They have lesser solute concentration. Because of these reasons, the water moves from guard cells into the other cells, and that cells become flaccid. Flaccid means they become flat. They are not turgid now. At night, when there is low solute concentration in guard cells, water leaves them and they become flaccid. In this form, both guard cells rest against one another and the opening is closed. Okay, you can understand that how the stomata that are present in the epidermis of the leaf are opened and closed. During daytime, the guard cells are turgid due to the presence of glucose. so that they attain a rigid board-shaped structure, and a pore is created between them that is called stomata, and that pores are open and exchange of gases takes place. But during the night time there is no process of photosynthesis, so the guard cells are in hypotonic condition and water moves out of them so that they become flaccid, and the pores present between them is closed. You can see the diagram in front of you in which both conditions are displayed. On right side you can observe the diagram of guard cells which are in turgor position. Turgor means they are rigid structure and the pores between them are open, that are called stomata. And on left side you can see that when the stomata are closed. So that basically the turgor of cells is responsible for maintaining the shapes of non-woody plants. बेसिकली टर्गर क्या करते हैं, वो प्लांट्स को रिजिड करते हैं और जो नॉन वुडेड प्लांट होते हैं उनकी शेप्स को मेंटेन करने के लिए हेल्पफुल होते हैं. नाउ वी आर going to discuss the application of knowledge about semi-permeable membrane. I have explained earlier that what is semi-permeable membrane. Semi-permeable membrane is a membrane which allows some molecules to pass through them and larger molecules cannot pass through them. Now we are going to see the application of this membrane in your daily life. You are familiar with membrane-based filtration systems that are present in our houses for filtration of water. That filter system is basically on the concept of semi-permeable membrane. A membrane is present in the filter. When water is passed through the filter, then the larger impurities are removed from the water. So basically the knowledge about semi-permeable membranes is applied for various purposes. We know that semi-permeable membrane is capable of separating substances. Okay, when we are talking that some molecules are cross the membranes and some molecules cannot, it means that it is responsible for separating the substances, so that it has vast applications in laboratory. We can use it for the separation of bacteria from viruses, because bacteria are larger in size than viruses. Bacteria cannot cross the semi-permeable membrane. So if we have a mixed culture of bacteria and viruses, we can separate it with the help of semi-permeable membrane. Some membranes are artificially synthesized, which are used for the separation of bacteria from viruses. Because bacteria cannot cross a semi-permeable membrane, in advanced water treatment technologies, membrane-based filtration systems are used. In this process, semi-permeable membranes separates salts from water, which is against the osmosis process, that is the reverse of osmosis. Now we will discuss our next topic, which is filtration. Filtration is basically a process in which molecules are forced to move across the semi-permeable membranes. Okay, the molecules that cannot cross the semi-permeable membranes, we can force them to cross the semi-permeable membrane with the help of water or blood pressure. Okay, now I will read the definition of filtration. Filtration is a process by which small molecules are forced to move across semi-permeable membrane with the aid of help of hydrostatic pressure. Hydrostatic pressure means water pressure or blood pressure. So students, you have understand the definition of filtration. What is filtration? Filtration is a process by which we can move the small molecules from semi-permeable membranes with the help of water or blood pressure. Now we are going to discuss the example of filtration. In the body of an animal, blood pressure forces water and dissolved molecules to move through the semi-permeable membranes of the capillary wall cells. It means that blood pressure जो है वो एनिमल्स की बॉडी में वो फोर्स करता है वाटर और डिजॉल्व मॉलिक्यूल्स को कि वो सेमी परमिएबल मेम्ब्रेन्स को क्रॉस करें सेल वॉल्स में से. In filtration, the pressure cannot force large molecules. Larger जो molecules हैं such as proteins to pass through the membrane pores, means proteins cannot cross the semi-permeable membranes even with the process of filtration. First of all, proteins must be break into the smaller molecules that are called amino acids. Then it will able to cross the semi-permeable membrane. So the students, in this lecture, you have to learn the opening and closing of stomata, application of knowledge about semi-permeable membranes, and then definition and example of filtration, so you can easily solve the question in your assignments. We will meet in the next lecture. God bless all of you.